It is my pleasure to welcome you to our monthly lecture series. We should start by noting that the land we're on is unceded land from the Wiyot people. And if you know the history, it got pretty ugly in the 19th century, a big massacre in 1860 and so on. I never knew that. I've lived here for 30 odd years. I did not know the history. And so I like to talk about it as dark as it is. The land is unceded. They are still here, participating in what goes on here in the Bay and have a stake in it. Let's see. I wanted to point out that we have two scheduled speakers coming up in January. A young man named Francisco Saavedra. He's a Yurok tribe member studying forestry. I heard him speak at a convention for California naturalists. He's a very effective speaker, very much worth attending. In March, just last night, I was here for a lecture on butterflies, and the speaker was so good, just totally kicked my butt. I twisted his arm, and he's going to come back in March and talk butterflies.
Can you give us the dates?
It's always the first Wednesday.
Okay.
Yeah, that would have been a good thing to write down, actually. Okay, so this speaker on butterflies, Clint Pogue is his name. He is part of California Fish and Wildlife Service, and that's his day job. His hobby is president of the Butterfly Association for the United States. So this guy wakes up in the middle of the night bringing butterflies. Like, yeah. Anyway, the talk was very effective. If you like nice, crisp organization and expertise, he's the guy. But you're not here for that tonight. Tonight you're here for our friends, the little tree frogs. And there's one now.
Yes.
Last week I was here, and just as I was coming up to the door, the little tree frog was knocking across the deck there, out to get out into the bushes. Our speaker tonight is Lindsay Mountz. And
hello.
He has just finished a project on them. And is it a secret that you just got accepted into the master's program?
It's not a secret. I was going to announce it to the masses this evening. Yeah.
All yours.
Thank you. All right. So, as we know, my name is Lindsay Mountz. I am currently an undergrad in the wildlife department here at Cal Poly Humboldt, and this project was advised by Dr. David Sin, who's right there. Thank you, sir. Yeah, I just got accepted into the master's program in Dr. Dan Barton's population ecology lab. So the work he and I are going to do is pretty loosely based on what David and I have done this semester for this honors thesis. And I guess let's do it. So we did already do a land acknowledgement, so thank you so much for doing that. The Cal Poly Humboldt land acknowledgement basically says that we are located on the unceded lands of the Wiyot people. We do honor them and all neighboring tribes by our ongoing commitment to developing trusting, reciprocal, and long-lasting partnerships. And of course, we would like to extend our sincere gratitude to all of you guys here at FOAM. This project was partially funded by FOAM through a grant, and that money was used to purchase supplies that we'll later talk about, some audio recording units, and basically just allowed us to make this project actually happen. So thank you guys so much for that. We also extend our sincere thanks to Dan Barton, Rob Blank, and Antoine Sherry, as well as Kelly Robinson, all from the Cal Poly Humboldt Wildlife Department, for their experimental design advice, technical assistance, field equipment allocation, and just general guidance. It is much appreciated. And I would like to extend my personal gratitude to Dr. Davidson for allowing me to have this opportunity, for advising me throughout this semester-long project and pushing me to, strive for greatness in my academics. So yeah, thank you. Thank you. Say the same. I would also like to thank my many peers, many of you who are here tonight, just for your ongoing support, for being a shoulder to cry on when things got tough throughout this project. I would also like to thank the Copper Mountain College Foundation for their ongoing financial generous support. The Copper Mountain College Foundation is back home for me at my community college that I went to prior to transferring here. I have a scholarship through them, and it has allowed me to really focus a majority of my efforts on developing myself academically and professionally. And because of them, I am able to go to college and not have to worry about being in debt or working countless hours to support myself through my bachelor's and my master's. So, basic little rundown of what we're going to be getting into this evening. First, I'm going to start off with the background, so just natural history of Pacific tree frogs themselves, Himalayan blackberry, and non-native species as a whole. So one of our big questions in this project was, are non-native species always a bad thing? Then we're going to move on to the actual study that we conducted. And overall, we're going to go over why we chose the particular methods that we did and how we accomplished our major objectives. And finally, we'll go over just some brief results and implications because I am still developing them. But we'll just go over what we found, how do we interpret our findings, and what these findings suggest in the broader context of overarching wetland conservation. So, I'm sure many of you have seen a lot of these species before. These are some pretty prominent non-native species. We have English ivy, Himalayan blackberry, kudzu, European starling, feral pig, and feral horses. So there is a distinction between non-native and invasive species. And depending on who you ask, there is quite a bit of leeway in those definitions. But for our purposes, we are going to address Himalayan blackberry as a non-native species. And
There for a second, do you think you could share your slides?
share them?
Yeah, right now they're not showing up on for the Zoom participants.
Oh, I'm so sorry.
Yeah, no worries. Sorry to interrupt.
It's all good. That's
Thank you
totally
so
my
much.
fault.
Yeah, no worries.
Let's see. How do I do that? Someone help. The green. No. I need help. I don't know. I don't. It was just under more. Okay.
Oh, Sarah.
Gosh.
Okay, great. And,
Excellent.
thanks, Jen, for jumping in and
Yeah.
advocating for the Zoom
Thank you
people.
so much. I would have just gone for an hour without knowing. Okay. Share at the bottom.
Supportive audience.
Gosh, thank you all so much. You're helping me develop my professional career. Okay, can we see the slides now? Are we solid?
Oh, yeah, don't forget your face got on, unless that was already.
Yes,
Okay.
it was. Thank you for pointing it out, though. Much appreciated.
Yeah, it looks
Right.
good on our end. Thank you.
Amazing. All right, so sorry, Zoom audience. My bad. I'm just gonna tuck this away forever. Oh.
Just a little nice bar on the upper, yep.
Okay. Okay. That's fine. That's fine. Is anyone bothered by that? Okay, great. Thanks for being good sports about it. All right. Anyways, some differences between non-native and invasive species, like I said, truly depends on who you ask. Some might change their definition of a non-native species based on the time frame. So, was this species brought to this native ecosystem 10,000 years ago or 10 years ago? Some people might think it's not worth it to make the distinction if it's so long ago versus more recent. And geographic distribution is a major one as well. Some species may be if it's a California native species, but it's causing destruction in a region of California, does that make it a non-native or an invasive? And also level of destruction. So some species may be non-native, however, they aren't particularly destructive to native species. And that's something that we definitely looked at here with Himalayan blackberry. So I just wanted to briefly go over those definitions so we understand the two terms. So how do invasive species really spread? For the most part, it's humans. One really prominent example is the Columbian Exchange. So there were a large number of invasive species from Asia, Europe, etc., that were brought to the Americas intentionally on ships. So we have horses, pigs, cows for those livestock purposes. But we also have some unintentional consequences, some unintentional invasive species like our diseases, like bubonic plague, that were also brought over. So some non-native species introductions are not facilitated by humans. Wind or water currents can contribute to the spread of invasive species. If you have a migratory bird that is flying through and a huge storm sweeps them out, and they kind of just decide, hey, it's not so bad here, let's set up shop. However, these are aided by human infrastructure, so by ship routes, etc., and also climate change. Climate change, as most of you know, I'm sure, is highly attached to humans. And climate change can cause many species to alter their natural distribution. So for example, with mosquitoes and avian malaria, mosquitoes are essentially being pushed higher in elevation because lower elevations are becoming unsuitable and higher elevations are opening up in suitability. So that natural distribution is changing and it's causing a lot of problems for passerine species. So our edible invasives. Himalayan blackberry is a really iconic, prominent edible invasive in our area and across most of the West Coast here in the US. And it is native to Near East of Asia. I put in parentheses Eurasia because it's slightly argued whether the plant originated in parts of Europe as well. It's not fully known the explicit origin, but places like Iraq, Saudi Arabia are truly where Himalayan blackberry is known to originate. And it is highly concentrated on our Pacific coast here, all along California, Oregon, Washington. It's sporadically peppered in through the East Coast as well, but we tend to have the more Mediterranean climate that is preferred by this plant. And it was brought to the US by Luther Burbank in 1885, so it was an intentional introduction. He brought seeds from India over to Santa Rosa, California here, and it has just spread like wildfire ever since. It is also established in the wild on all continents aside from Antarctica for obvious reasons. And it was thought to have originally been introduced globally for commercial use because of its fruits. So small-scale cultivation was the primary driver for bringing this plant here. However, it escaped cultivation and established itself in the wild. And rubus in our area. Rubus armeniacus is the Latin binomial for Himalayan blackberry. So there are some times where I may refer to it as rubus, sometimes I may refer to it as Himalayan blackberry, just so we understand that distinction. The reason why it is so widely recognized in our area is number one, they're absolutely everywhere. I mean, you can't really go to a natural space and not see rubus at this point. They're useful and tasty. I mean, picking blackberries and making a yummy warm pie is so fun. It's so great. Everyone loves it. And they're also painful. If you're ever hiking on a trail and you go through a thicket of blackberry brambles, no one's going to have a good time. And I also thought it would be fitting to include the Wiyot name for our blackberries as well. And here we have a picture that kind of shows the distinction between our Himalayan blackberry flowering plants and the California blackberry. So It is a bit difficult to tell the two apart, but it is known that the Himalayan blackberry has largely extirpated the Pacific or trailing blackberry that is native to our region. All right, and Pacific tree frogs, the other star of the show. Currently, their conservation status is least concern. They're very stable, and they were previously classified as Hyla regilla. They're currently classified as Pseudocris regilla. Hyla regilla was branched off into 3 separate species that all span the length of California. However, our special Pseudocris is in blue up here in this tiny little region where we are. And they do serve as important prey and pest control across the board. They are prey for some large bird species like great egrets, which we have plenty of here at the marsh. They are pest control effectively. They eat flying insects. So if you are a farmer and you grow crops, these little guys are pretty helpful in eating the pesky little insects that like to live in your crops. Some conservation concerns related to this species include climate change. They are amphibians, so climate change, of course, affects the temperature, the precipitation, the occurrence of natural disasters, etc., that can really blow these guys out of the water, pun intended. And wetland delineation. Currently under our administration, wetland delineation is kind of changing overall. And for those of you who don't know what wetland delineation is, it's essentially just someone says what a wetland is. Who makes a wetland? What is a wetland? People can just say, this is a wetland, this is not. And under our current administration, sorry, that was laser runner. Under our current administration, environments like vernal pools, for example, that are very seasonal wetlands are kind of being taken out of the government's definition of what a wetland is. And those vernal pools are major important ecosystem structures for this species because they use them for their breeding behavior. They go to these vernal pools, they breed, they lay their eggs, the eggs hatch, they develop into adults, and they're on their way, and that cycle repeats. And they have very specialized breeding cycles. So they breed in the spring, and that's when the rain comes through, fills these vernal pools up, and we protect them because they're so important to amphibious breeding processes. However, current legislation is kind of pushing those guys to the back burner and not protecting them as much, which is, in my opinion, quite unfortunate. And the last major conservation concern that I wanted to add in here is disease. So there is one major disease that is affecting amphibians across the globe, which is chytrid fungus. Batrachochytrium dendrobatitis is the current strain, if you will, that affects frogs and toads. However, these guys are not largely impacted by that disease, which is great. Yes, I saw a thumbs up. Close relatives to the species are unfortunately quite impacted, which if we're able to assess habitat requirements or preferences for these guys, it may be possible to kind of extrapolate that information and use it as an overarching theme for our conservation principles for other amphibious individuals. There's also a disease, Riberoria on Datrey, I believe that's how it's pronounced. It causes them to, it's a developmental disorder essentially, and it causes them to form a third, sometimes even a fourth limb at their limb bud. So where there usually would be one leg, there can be up to 4 or even more limbs. And that's also something that should be addressed with these guys because it makes them far more susceptible to predation. And of course, that makes their populations go down, and then there's more frogs. And if anyone would like to answer, there's no pressure. Why might Himalayan blackberry be attractive to our local frog species? Does anyone have any ideas? Yeah.
The thorns keep away predators.
Totally. Yeah, that's a really good point. Yeah.
The fruit when it's rotting attracts insects.
Absolutely, it does. Yeah. I saw a hand in the back. Oh, awesome. Anyone else? All right. Yeah, you guys totally hit it. our ideas were that the rotting fruit would increase insect prevalence, and that would, of course, attract frogs because, ooh, hot pot, free food, I love it here. And like you said, I really like the thorns idea. Their prickles definitely can be a deterrent to predators. I mean, these guys are pretty hardy, as you can see in the top right photo. That is a large picea branch. And I don't know if you guys have ever touched like a pine branch. It's not very comfortable. And these guys just choose to be there. So they're pretty hardy. They can handle a little prickle or two. Whereas, a great egret might say that's not so worth it for me. They also provide a lot of vegetative complexity. So these are low-growing shrubs. They create a lot of cover on the ground, and they're extremely quick growing. So you can start out with a small bramble, and it'll expand over an entire plot, and basically you have no ground visibility. Yeah, 20 minutes, exactly. Might as well be. So that increased ground cover, of course, would increase the darkness under that vegetation. So it would decrease light penetration, which means maybe temperatures would be a little cooler underneath. Maybe moisture would be retained a little more. So that was kind of my thinking process in the beginning of this project. And that's why I hypothesized that this vegetation would be associated with higher densities of flying insects, higher soil moisture under the vegetation, and lower overall soil temperature underneath this vegetation. So our study area, as some of you may know, was at Gannon Slough, and that is not part of the marsh. However, it is part of the larger Arcata Baylands project. So it is a wetland ecosystem. It's a very essential wetland ecosystem. And I'm sure some of you may know that over 90% of California's historic wetlands have been destroyed, largely due to agriculture and urban development. So it's very crucial that we understand wetlands as an overarching large system of ecosystems kind of blended together in this little web. So it is a 2.42 kilometer square area, and it's characterized mostly by low-growing grasses atop a flat middle ground, and it's bordered by mixed vegetation. So on the right photo, that tree line is the western tree line. So that is the one that faces the 101 going north. And it is largely characterized by pine trees, rubus, and spreading rush. And then here on the left, you can see a bit of the actual slough running through there when it's inundated. And this was mid-October, so it was pretty nicely bolstered with some water there. And that tree line over there is primarily red alder trees, which are a native tree species here in Humboldt County. And that tree line is eastward. So it is toward, there's like a little league field over there in the CHP office. That's where that tree line kind of borders. And there also is another sort of ravine behind that tree line. So there are some water sources kind of sprinkled throughout the slough. And the study did span over about 2 months. So from mid-September to mid-November, there was actually sampling taking place. And part of our experimental design, we used PVC artificial refugia based on a previous study done by Myers et al. In 2007. We, for our study, had 60 total PVC pipes. So here on the left, I have a photo of that actual setup. We used 10-inch long PVC pipes that were 1.25 inches in diameter. And we had a socket cap on the end. So it would hold in water. I thought you would need like rubber cement or something, but they're pretty watertight. It was really neat. And we drilled 2 holes in the bottom about 1 inch above that little socket so that excess water could drain while it rained. And for certain vegetative types, if it were like spreading rush, for example, or pretty underdeveloped blackberry, we would use a rebar stake. So a 6-foot tall curved rebar. And we would tie string to the rebar and hook it through a hole in the PVC so it would be pretty nice and secure for the frogs there. And as you can see, they didn't mind them too much. And in order to select our sampling sites, we had 20 total sampling plots within our sampling site at Gannon Slough. And in order to select that, we did a systematic random sampling design based on Sylvie 2012. So in this, we went into a GIS mapping software and we drew a polygon around the Gannon Slough wildlife area. And we said, hey, generate a random point. And it generated a random point. So I went out there on the ground to that point and I said, hey, where's the nearest tree or bush or what have you? And that's where we put our first pipe. And then from there, we went 5 meters, put another pipe. And then we went 5 meters, put another pipe. Great. Now we have 3 pipes, and that's our first established plot. So each plot was 10 meters long and 2 meters deep, and there were 20 total plots. So each plot was 50 meters apart, and that spanned across pretty much the entire Gannon Slough wildlife area. It was about a 2-mile hike each time I went out there, which was a lovely workout, in case you guys are interested. We had 3 pipes per plot, like I said, and about 2 inches of water were added to each PVC pipe to attract these amphibious individuals. Voice crack, sorry. And these are our plots along both tree lines. The red markers indicate the initiation of the plot. So the first encounter point that you get when you're walking south along the tree line. And then the green markers indicate the end of a plot. And for our frog counts, we did visual observations twice a week for a period of 5 consecutive weeks. So I went out to the slough and I looked at each of the pipes and I basically just decided, hey, is there a frog inside this pipe or is there not a frog inside this pipe? It was recorded. I also recorded abiotic measurements like weather, 24-hour precipitation, etc. And I only counted frogs that were physically in the pipe at the time of me seeing it. So if I saw a frog on the ground in the vegetation hopping around underneath the pipe, that didn't count. And during these counts, I did some basic pipe maintenance. So sometimes there would be debris in the pipe. I cleared the debris, put fresh water, and we were good to go for the next round. And just to preface, there were no frogs handled during the study. It was not necessary. And according to our IACUC permit, it was not part of our project. So we just decided there's no use in putting frogs through that stress. Might as well not handle. And we used, based on our visual observations, we used cumulative frog counts for each plot in our data analysis. So for each of the pipes in each plot, we did those individual counts, and then we added up those counts for each plot, and that's what we used in our analysis. So our null hypothesis revolving around frog counts and vegetative type stated that there is no significant difference in frog counts across vegetative types. And our hypothesis, which is called the alternate hypothesis, was that Himalayan blackberry facilitates larger frog populations. So we would see more frogs in areas that had more blackberry or any blackberry period. Another thing that we did measure was food availability. So you guys that said the insects, you were totally on it. We looked at food availability with mostly mosquitoes and flies, just basic house flies. And we used 2 double-sided sticky traps per plot. So they were LEAQU brand PLGEBR, double-sided yellow sticky traps. That was a mouthful, thank you. And they were deployed for 72 hours each. And total bug counts for each trap were averaged and recorded for data analysis. So this is a photo of one of the lovely bug traps after it had been out for 72 hours. And for these bug counts, our null hypothesis was that there's no difference in flying insect populations across the vegetative types, so native versus non-native Himalayan blackberry, or native vegetation versus non-native Himalayan blackberry. And our hypothesis, which is the alternate hypothesis, stated that rubus facilitates larger flying insect populations, and increased food availability may contribute to larger frog populations, like we previously discussed, because they find this honeypot of food and they just don't want to leave. And we also measured some pretty important vegetative characteristics throughout the study. The first one was soil moisture. So we used a Vegetronics soil measuring meter, which can measure luminosity, it can measure soil moisture, soil temperature, and that's about it. Those are the only features I used. So soil moisture, we did 3 readings per pipe, and then we averaged those 3 readings and took it as a plot value. So each pipe got 3 readings, and there were 3 pipes per plot, so there were 9 readings per plot. And for the soil moisture, our null hypothesis is that there's no significant difference in soil moisture across vegetative types, and our hypothesis was that Himalayan blackberry would be associated with heightened soil moisture due to that vegetative complexity, creating more shade in the understory. Similarly, with soil temperature, we did the same 3 readings rule per pipe, and then we took an average for each plot. And our null here again is that there would be no significant difference in soil temperature across the different types of vegetation. And then the alternate is that Himalayan blackberry would be associated with reduced soil temperature, again, because of that shade due to the increased vegetative complexity. And then finally, we also estimated blackberry cover. So this one was a lot less technical, if you will. We did a simple binning method. So essentially, you go to your plot, you look, and you say, that looks like about 10% to me. And for us, in our study, we rounded to the nearest 10%. So we binned to the nearest 10%. And this figure can show a pretty decent description of that binning method. So they have a quadrat in each frame, and as you can see, they binned to the nearest 10%, I believe. So yeah, that was our blackberry cover method. And our null hypothesis was that there would be no significant difference in frog occupancy across areas of different percentage blackberry cover. And our hypothesis was that there are more frogs in areas with higher percentage blackberry cover compared to areas with native vegetation, or with less blackberry cover, excuse me. And vocalization data. These are called ARUs. They are autonomous recording units. And these are what we used to record our nighttime vocalizations in each plot. So we used 1 ARU per sampling plot to collect vocalization data at night. And this was done over a period of 7 consecutive days in each plot. And each night recording session was 4 consecutive hours of recording from 8 p.m. to 11:59 p.m. And that's just because we're aware of the fact that frogs vocalize primarily at night. I'm sure during the springtime, you've all gone to bed and heard the lovely chorus of Pacific tree frogs. And for analysis purposes, we listened back to the audio recordings for each plot to identify Pacific tree frog vocalizations in order to assess occupancy for that plot. So what that means is we listen to the files, and if we heard a frog vocalization, great, that means there's frogs there. We can stop listening. We're just going to record a 1 for occupancy or a 0 for absence. So if we didn't hear any frog vocalizations for a plot, they got a 0. And maybe this might work. It's a frog vocalization. Oh, it's not going to work. I broke it. I'm so sorry. Well, you guys are just gonna have to find out for yourself.
Can you mimic it?
It's like a. That's my best impression. That was pretty good, right? That was pretty good. Thank you. Thank you. Thank you. And anyways, so her results. This is what everyone's been waiting for. So for treatment versus frog count, as you can see in our bar plot, we didn't really find enough evidence to accept our alternate hypothesis, which means we failed to reject the null, which means there's no significant discrepancies between vegetative types in terms of frog counts. And for these data, we conducted a Shapiro-Wilks test for normality, and our data failed that test of normality, which means we should do something like a Mann-Whitney U test, and that value showed that we failed to reject the null. Similarly, for treatment versus food availability, so that's do we have native vegetation or do we have blackberry, and how that is connected to the amount of flying insects there. We also failed to reject our null. We also did treatment versus vocalizations, and for these data, because they're both categorical, we conducted a chi-square test, and that is best represented through a table. However, I just figured I would tell it to you in this case. And again, we failed to reject the null based on our p-value, which is above 0.05. And finally, for treatment versus soil moisture, there was again no significant statistical difference between vegetative types. And in other words, we failed to reject the null. So what does this really mean? Is this relevant? Is this reliable data? Our data show that there's not a significant amount of evidence to support the claim that Pacific tree frog populations are facilitated by Himalayan blackberry plants. In other words, they don't really show a distinct preference for this non-native species. However, this is not by any means a comprehensive study. So little old me doing a 2-month-long study at one sampling site isn't really a great representation of all Pacific tree frogs. And many improvements can be made to execute a series of more conclusive studies that would allow us to draw more discernible conclusions. And in order to do something like this, we would want to, of course, expand our number of sampling sites. So instead of just one site at Gannon Slough, we could expand to Gannon Slough, Arcata Marsh, Wright Refuge, Faye Slough, etc. Having more populations represented would give us a better relationship illustration. We can also extend our time period. So instead of doing a short 2 months, we can do multiple years, multiple populations over multiple years to get a better idea of the true representation. And in the off chance that our results are truly accurate to the overwhelming majority of Pacific tree frogs altogether, then I would say the relationship between Himalayan blackberry and Pacific tree frogs is essentially negligible. They don't have any sort of detriment associated with them. They don't have any discernible benefit associated with them. Thanks, y'all. Yes.
I hear bullfrogs are a real problem for other frogs,
Yes.
predation. Did you find any in this study area?
I did not. No, I found California red-legged frogs, which are a bit larger as well. But those guys are stable, and I don't know of any negative interaction between those two species either. Yeah, thankfully. Yes.
Wondering about the sharpness of the thorns.
Yeah.
And the length of the thorns. I had tree frogs, which we call forest frogs.
Yeah, sure.
They're giant blade.
Mhm.
And they have little tiny, teeth on the edge of the, but nothing like this. And
Yeah.
I'm wondering if the thorns wouldn't be prohibitive
Mhm.
for the frogs and maybe doing a different non-native invasive
Absolutely a really good point, and I agree with those sentiments. I know that from just talking to community members and from my own personal observations, we have seen Himalayan blackberry associated with frogs in general. I mean, we've seen them on brambles, we've seen them in the understory, etc. So I don't think they're terribly bothered by the thorns or prickles themselves. Like I said, these guys are relatively hardy. I mean, in this picture as well, this guy's just hanging out on a bramble, which I don't really know. I personally wouldn't be doing that. Uh, but no, I do agree. I think looking at other invasive vegetation would be a really good expansion upon a project like this. Yeah.
And do you have any comments about management of the Himalayan blackberry? I mean, one thing we do at the Marsh is cut it down in certain places. And it sounds like your study says we don't have to worry about homes for the frogs as much as we might, but
Sure.
Is there anything that you could tell us about time of year or
Totally.
how to watch out for them so we don't nail them all or
Yeah.
cut it out?
I know for sure from what I read that the Himalayan blackberry is outcompeting our native species of blackberry, so the Pacific blackberry, also known as trailing blackberry, which can be a problem because there may be obligate species that rely specifically on our native species of blackberry, and also it's kind of an overarching theme that we want to preserve our native biodiversity just because we appreciate what it is and what has historically been here. In terms of time of year, I think really when it's fruiting and flowering, that's probably the worst time of year to try and manage it. You want to, get it before it starts to fruit and flower. So as a means to prevent it from spreading while you're trying to get rid of it. In terms of vegetative management overall, I think because it is a non-native species, it might have the capacity to just overall outcompete quite a few native vegetation species, which is not necessarily the greatest for our purposes.
Since you couldn't find a correlation between frogs and blackberries, do you think that maybe expanding your sampling scheme to include frogs, maybe not in pipes, but in that vegetation could help create more of a positive or negative correlation? Because it was kind of set through kind of a narrow lens as only frogs in these pipes, and there could be more to it, obviously, if you expanded your sampling scheme to include that.
Absolutely, yeah. I think that would be a great way to expand our sampling scheme. For the pipes, it was just based on a prior study that was done that showed they kind of like the pipes, So that's why we decided to go for this safe bet. And it's also fairly accessible for us as humans because, rummaging around deep in the thickets of some blackberry brambles is not the easiest work to do. But I do agree. I think just visual occupancy might be a really good way to take another approach at this. But also, I can think of some drawbacks like replication of individual counts, etc., that might kind of skew some things. Asher, yeah.
You've got your audio recordings too.
Yes, I do.
Why the water in the bottom of the pipe?
Just because they're amphibians, they prefer to be in wet spaces. So I think the water is kind of just a cherry on top for them. It kind of allows them to be a little comfortable in there. Izzy, yeah.
I'm assuming like each plot was classified as either like with blackberry brambles or not.
Yes.
Was it just based off of like presence of blackberries like inside the plot, or was there like a range outside of the plot where you like noted if there were like brambles or not?
So the only blackberry I counted was inside each plot. So based on that systematic random sampling, I lined out my plots, and what was in, what was out was out. So if we had a blackberry shrub that was partially in the plot and partially out, I only counted the percent cover that was actually inside my defined plot. Yeah.
I have a couple questions.
Okay, great.
My first one is, if you had the ability to like further this study for X amount of time, more resources, with the knowledge that you have now,
Totally.
where would you want to take it? Because I remember when I was finishing my honors thesis by the end of the semester, like looking at my results and stuff, like it made me really curious, like, oh wow, these results make me want to go about it in this way.
Totally.
And your results being like inconclusive or insignificant, like, does that make you think like, okay, well, this is probably just the answer what I'm looking for, or maybe I want to go about testing it in a different way or
Yeah.
different location?
No, totally. I absolutely have gone through that thought process, and I think a bit of what we've talked about this evening has touched on that as well. Really just expanding the number of sampling sites, and then I can also chunk down plots within the multiple sites and do a similar survey method with the frog counts, and possibly even like foregoing the pipe idea or keeping the pipe idea for part of the study and then doing just visual counts for another part of the study, or expanding to different species. Do different species of frogs have different preferences, or do all frogs have a preference, or what have you? I definitely think that upping the number of sites, the number of people on the ground, that would help a lot, just because it's quite a laborious study. Extending the time period is a really big one as well. I think being able to do this over the course of at least 2 or more years would be a better way to go about it. Yeah.
Are they strictly terrestrial? How much time do they spend in the water?
As far as I know, they are aquatic breeders. So they go back to the water for breeding, and their eggs are aquatic as well. I believe most of their time is spent terrestrially, but I've also seen adult Pacific tree frogs just hopping around in little pools of water on the ground. So I'm not entirely sure about like an exact time breakdown, but I know definitely they spend quite a bit of time in the water during the spring. Yeah.
This is sort of a question like that. There are a lot of Pacific tree frog lovers here at the Marsh, so this is
Wonderful.
a question for you, just
course.
having spent a lot of time with them and looking at that great picture. Have you learned, like, we're always curious about the green ones versus the bronze ones, versus
Sure.
the half bronze ones.
Sure.
Do you in your sampling keep track of any of that?
That is an amazing question. That actually was one of the original thoughts for this study. Was we were going to identify these individuals. We were trying to think of a way to do a sort of mark recapture or identification of different individuals, so we wouldn't count replicates in our pipes. So if we could make a little color scheme or a pattern recognition palette of some kind to be able to keep track of individuals, that would be amazing. I think that's a really awesome idea. And I think it's a really understudied area of wildlife in general. And that was just something that would be a bit too laborious on top of the study, but I do really like the idea. Yeah.
Just percentage-wise, from the ones you were looking at,
Mhm.
80% all green, how to do?
I would say there were fewer all green ones in comparison to those that were kind of half and half or mottled with like a rusty color. There were some that were entirely like that dark rusty brown color.
Yeah.
There were some that were entirely brown. They had no green on them at all. They're really variable, and we do believe it has to do with the vegetation that they're surrounded by. I don't really know though.
And then any pointers if you hear them, how you might then find them?
That's very tough. That is very tough because they're so elusive. They're so good at hiding and blending in and kind of tucking themselves into little nooks and crannies. I think really just being patient, because I mean, there were so many times where I was out there walking through the brush, and all of a sudden this frog just like hops up as tall as me, and I didn't even realize it was there. It's just one of those things where you have the magic touch or you don't, and I don't think I necessarily do, but I think Asher's quite good with amphibians.
I was gonna ask, there's what months what was the time frame?
Time frame was September through mid to end November. So it was not during their height of activity. It was their off season, if you will.
Because that could feasibly affect that too. 'cause
Absolutely, yeah.
change color later in the year, right?
Yeah, definitely. That was another part of the study that was a little tricky to kind of work around because they breed in the spring. They're more active in the spring because they're trying to have babies. And it's a little tricky when they're a little less active. They're ectothermic, so they're not going to be moving around so much when it's so cold. Um, I think that was also something that might have affected our data as well. Yeah.
I'm curious about why you chose to study it with the Himalayan blackberry as opposed to another invasive species.
So the Himalayan blackberry is just something that is so plentiful. It's so prevalent here in Humboldt County. And it's we knew there were frog populations at Gannon Slough based on personal communication with another wildlife faculty member who had seen those populations before. And David and I, he taught a class in which we did a very similar study to this, where we knew there were frogs there and we knew there was blackberry there. And I think just the fact that it's such a prominent invasive, it's so accessible. That was something that was really alluring. And also, a lot of people that I just talked to in my degree program have said, oh, frogs love blackberry. Frogs are always in the Himalayan blackberry, just because a lot of people commonly see the frogs using blackberry as habitat.
Okay.
Yeah,
A couple questions.
totally.
Really interesting. The first one is about, were you visiting the sites at the same time of day through September to November, like starting at 9 and then moving your way south?
For the most part, yes. So there were my first survey day of the week. I went out there around 1:30 p.m. each survey week. And then my second survey day of each week was around 4:30 p.m. And I always started at plot 1 and made my way down to plot 20 and then walked out. Yeah.
And then I know you didn't touch the frogs, but I'm
Yes.
curious if you noticed anything about the sizes, because I've also seen them. I walk a lot around the treatment ponds in the Greenville area. Yeah, they love the blackberry, but they're always like on the leaves. I hardly ever see them on the stems like that.
Yeah.
But it's like you see the little ones, you see big ones. Did you see anything different like that, or just feel like they were sort of evenly distributed?
I think they were for the most part, pretty evenly distributed. I did see there was a little population of really teeny tiny brown ones that really liked plot 3. And I just kept finding the tiniest little brown ones in there, all kind of huddled in together. It was honestly kind of cute. But in terms of variability across the entire sampling site, I think it was pretty consistently distributed. Yeah. Yeah.
Do you think that you'll expand the vegetation survey as you expand the study, or if you get the chance to?
If I could, I think it would be a really nice addition, definitely. I mean, Himalayan blackberry is very accessible and it's everywhere, like I said. But I don't know what other species would be good candidates. I'm just not super well versed on, what people's observations have been, in their day-to-day life. But
It's just interesting because a lot of areas in the marsh are adjacent enough to cattle and such.
Yeah.
That there are a lot of other introduced species, obviously, that are less pronounced. It's grasses. It's
totally.
other things like that are easy to look over. But I
Yeah.
wonder if you could add that into the study or.
Absolutely. I think that would be an awesome idea. And I also know that the Gannon Slough itself is leased out to cattle ranchers to graze their cattle. And there are a lot of those low-growing grass species that kind of bunch up really nicely. And I did see quite a few frogs kind of just hunkered down in there. So I agree with you. I think the grass species would be pretty interesting to look over. Yeah.
I wondered what the effect of when we have extreme droughts for several years has on the populations, if you include just as a summary guess.
Yeah, I mean, of course, with any amphibious species that relies on water, any drought is going to definitely impact their viability. It's going to impact eggs can dry out, individuals can dry out, and that can be really stressful on the individual themselves. And that general stress can also reduce their immunity, so any diseases that they might encounter can have a larger effect. Yeah, I think drought could also have to do with their activity levels. Yeah.
Well, kind of a stupid question, but what are
No
they-
stupid questions.
So what are they during winter? Like, is this the coldest that it gets for them, and do they just get like slower and less active, or do they like bury themselves in the dirt, or like do anything else?
Honestly, that's a really good question. I have not heard of them burying themselves in the dirt around here, at least, because we have such a moderate climate. And I know this is off topic, but with rodents, rodent species that frequently undergo torpor or hibernation don't really tend to do that here because our climate is so moderate and we have relatively warm winters compared to other places. So they're kind of just agitated out of that rest state. And I think it could be similar with our amphibians. I didn't really see any that were kind of burrowed under the substrate. I know that from my personal observations, it seemed like they were kind of just slowed down. They weren't as plentiful and moving around so much. Because again, they are ectothermic, so they kind of just rely on the sun and shade and whatever else can externally help them regulate their body temperature. Yeah.
Where does your study go? What happens to it when you're finished?
When I'm finished, I know there is one individual in the wildlife department who's also an undergrad that is interested in doing an expansion on this study for their senior thesis. So that would be a really interesting addition, I think, for someone to carry on the legacy and try and see what we can do to just widen our scope of understanding on this topic. And in terms of my results and my final product for this project, I do have a manuscript that I have to submit to the university and to Dr. Davidson. And there's also a poster session. There's a thesis poster session, December 12th, in the library at 4 p.m. 4 p.m., December 12th, second floor of the Cal Poly Humboldt Library. Be there or be square. Yeah.
What made you like interested in frogs and like inspiring you to study?
Oh boy, that's a crazy one. No, good question. Very wonderful question. So my first time ever seeing a frog ever was when I moved here, and that was less than 2 years ago. So I still consider myself extremely new to the world of amphibians because I had never seen a salamander or a frog or a toad or anything like that until I got my boots on the ground here. So I think it could just be like a novelty thing that piques my interest partially. I also think they're just really cool. They're super elusive, they're understudied, they're underrepresented in science as a whole. So I think, finding out more about them is.
They're awesome.
Yeah, they're adorable. They're so cute. They're so awesome. I think it's really important that we actually look into these guys and understand them better. Much appreciated. All right. Let's see if there's.
Can I give you a kudo?
Oh, please. Thank you.
You have been really excellent, I think, at accepting and hearing suggestions, questions, and all your responses have been positive, each person.
Oh, thank you. I try. I mean, there's no point in doing science unless you're willing to collaborate and encourage others. So I think that's a good way to be. Awesome. Thank you guys so much for coming out. I really appreciate it.
Do we know how many people were online?
I don't. Let's see if we can get back to Zoom.